Hand over to a different topic. Paul Darby from University College London is going to tell us about novel biopharmaceuticals and the formulation challenges thereof. So Paul, great, I can see your screen. Andrew, if you want to switch your video off, then Paul, is your video on?
It is. Should I turn it off?
No, leave your video on. It's nice to see you. I mean, sorry, just you hadn't shown up, but that's because you hadn't made a noise. So, okay, I'll hand over to you. So if you want to go full screen, we can see your screen. Great, thank you.
That work? Great. Okay, so I'm going to present some work on biopharmaceutical formulation, and it's in collaboration with Robin Curtis and Jim Warwicker, who are from Manchester, and they're both here today. So just a brief introduction. Biopharmaceuticals are a very rapidly growing sector of the pharmaceutical market, certainly in value, but also that they're in the top 8 of the top 10 best-selling drugs at the moment, and over 25% of new approvals are biological, so proteins and advanced therapies. They're getting more and more complex, which is because protein engineering and conjugation with other molecules, small molecules, RNA, etc., are all now possible, and that is making, while they're more potent and more useful as a drug, it makes the challenge of engineering them to be stable in a dosage form very, very much more complex. The problem, though, is that there is time pressures, as ever, to get a formulation, particularly as phase 1 clinical trial is going on. You have an initial formulation, and you might want to change that as you learn more about that formulation over time, because you've rushed into getting a formulation for phase 1, and then you get the shelf life over 2 years, which is what you need, probably as later clinical trials are about to take place. So you need to fix your formulation quick, so learn about your product, get the formulation done. That's extremely challenging because there's not much material, there's a lot of complexity in formulation, a lot of things that you need to stabilize against. We need to have the product stable, but it has to remain potent and also meet whatever conditions for delivery to the patient are required. So the right format to get it to the right place. Another challenge is that many of these therapeutics are increasing the concentration at which they need to be at, and this is increasing their degradation rates, their rheology, they become more viscous, for example, and also sometimes lead to phase separation, which are all problems that need to be solved. So I'm going to focus on aggregation. That's where we put most of our efforts, because that's the sort of #1 challenge, usually with these kinds of products. And getting any idea of predicting and controlling aggregation by any means, formulation or protein engineering, is very difficult. And that's partly because understanding aggregation is still really only emerging. And that's because some of the species, and there are many species in aggregation, they have such low populations, they don't hang around very long, so you can't isolate them to study them. There are also often multiple mechanisms going on at the same time, and that is very dependent on what conditions you put it in. So the formulation itself can change the pathways that are dominant. In terms of getting a formulation and beating aggregation down, the industry is sort of reliant initially in going into the first clinical trial material, which might be in a few months. They're dependent on kind of rapid screening approaches, so high throughput methods, experimentally measuring things like the unfolding temperature of proteins or the free energy of unfolding, colloidal stability in terms of the temperature at which they aggregate, and there are also, as Robin Curtis does, sort of measurements of protein-protein interactions in terms of B22s, for example, or KDs. So these you can do relatively quickly on many samples. And then also you have the potential to accelerate degradation, increase the temperature, agitate the sample, for example, and then assume an Arrhenius type extrapolation back down to the temperature at which you're going to store your material. Part of the problem with making using those methods and assuming they'll work on the shelf over 2 years is that aggregation is a very complex thing. There are many aspects of the protein structure on the left which can affect it. Conformation, the surface properties, sequence level features that cause aggregation prone regions, APRs, and then also on the right what you're putting in. So the excipients you put in, do they interact with the protein? Do they change the viscosity? Do they crowd the molecule? And all of those can impact on aggregation and also some other features like solubility and fragmentation. So it's very complex, and they all interact as well, just to make life easier. So one of our aims on top was not only to understand it, which was aim 1, but how can we then try to use this information and predict better formulations, particularly if time is against us, can we do it computationally to get an initial formulation that's likely to last the course into 2 years on the shelf at 4 degrees. And do the methods that we use, are they good enough? So to measuring melting temperatures and forced degradation, are they really predicting shelf life? How well did they do that? And then what other methods can we develop? And then if all of that begins to work, can we also engineer the protein, maybe through protein mutation or through formulation to get lower aggregation rates? And an additional aim was, what can we develop new excipients based on what we learned? So some of the work that Robin Curtis does in Manchester is around that area. So these are our objectives. Essentially, objective 1 was to collect lots of data on these different properties under different formulation conditions. Objective 2 was to kind of pull that all together using informatics and modeling to build predictive models and understand it better. And then we had an objective 3, which I'm not going to talk about, but there is a whole talk I could do on it, on analytical methods that we've developed as well to improve the way we characterize them. So work kind of began based on this sort of data, collecting aggregation rates over lots of conditions. So experimentally hammering out lots of data in terms of aggregation kinetics at different temperatures, pH and ionic strength, which are the 3 major things that tend to affect aggregation rates on top of what you then add to stabilize it in terms of excipients. We also have a similar heat map to these shown on the left, set of heat maps in terms of the melting temperature under the same conditions. So we measured all those and then essentially compared them. So this was all done, the data here, for an antibody fragment called A33, which is that protein structure shown up in the top right. The data is collected using chromatography methods to monitor degradation or loss of the monomer, the original protein. So early work was to compare, did the melting temperatures we measured correlate with aggregation kinetics? And essentially melting temperature we converted into the fraction at any temperature of which the protein is unfolded. So you've got the melting temperature Tm, but also at, say, the incubation temperature that you're measuring your degradation, what fraction of protein is expected to be unfolded? And that's the F, T on the x-axis of the plot on the right. And what we found was there isn't really much of a correlation once you drop the incubation temperature for your aggregation kinetics to sort of 23 degrees or 4 degrees, which is where you want to be. That's for your storage. And it really came down to, once your protein is very native, which means it's got a very low F value, then the unfolding temperature is not really the driving factor. Unfolding of the protein is not what's driving its aggregation, so it doesn't affect the kinetics. So that we learned pretty early on. We were then wanting to mechanistically get behind what is driving aggregation at a structural level. So we went out to Grenoble, in this case, to do some small angle X-ray scattering on their synchrotron. And here, essentially, we took one of the conditions at temperatures at 23 degrees C and explored all the different pH and ionic strengths and got scattering curves. And you can see the initial measurement you can get is the radius of duration. And we could see straight away that there was a pH dependence of that radius of duration, showing that the radius was slightly larger, about 7% larger at the lower pH. So that's that plot in the middle. On the right is when you correlate that radius of duration against the aggregation kinetics for the different pHs, and we can see that there was a definite switching on of aggregation kinetics at the low pH, which we knew, but that also was the same conditions where the radius of duration was slightly larger. So it looked like there's a structural change happening at the same time as the aggregation kicks in. So we looked at molecular dynamics, first of all, because it's easy to sort of access at these different pHs, and we could start to see which regions of the structure were becoming more expanded, more unfolded, and certainly, yes, at low pH, there were regions, particularly in what's called the CL domain of the Fab, that were more solvent accessible. That's this bit that kind of stuck out just at the end of that video on the right. But you can do more with your data than just that. You can take the molecular dynamics and you can take the SAXS data, the small angle X-ray scattering, and combine them. So essentially look for, within all of those structures in the molecular dynamics simulations, which ones best explain the SAXS scattering data. And there's nice tools you can use to find which structures are best fits. And then you take the best 10 or the best 20 and you overlay them. And we could see that essentially these are a series of best fits for each pH. So pH 7 in the light blue, pH 5.5 in yellow, and pH 3.5 in the magenta color. And we could see very subtle changes in the CL region where the structure was moving. And it was really quite small, but that is what explained the 7% radius of duration change. So it was the CL domain that was having some displacements happening. We wanted more proof. It's always good to get orthogonal information. And so we turned to an approach using single molecule fluorescence. And here we use something called FRET, so forced to resonance energy transfer, where you double label your protein with two different fluorophores that accept light at different wavelengths, and one will excite the other when it emits. So you have a donor and an acceptor. And how far apart those two fluorophores are determines how much fluorescence you get out of the acceptor, because you're only exciting the donor, and it's dependent on the 1/r⁶ distance of q to the power of 6. And so you can get a very quick sort of read off at single molecule level. You get a histogram of all the different conformations that exist in your solution. We looked at those at the different pHs, and we could, I'm not going to go through this in detail, but we could indeed see that when we labeled it, so we put the labels at different positions, when we labeled on the CL domain, then we saw a shift in the FRET efficiency, which told us that those probes were getting further apart, and it was exactly correlating with what we saw by SAXS and MD simulation. So we're absolutely certain of the region of the protein that was now beginning to unfold as the protein aggregated. Looking at those in more detail, we can look at sequence-specific motifs using, there's a whole series of calculations. We actually took, there's about 15 online calculations for aggregation propensity dependent on sequence of the protein. And you can get a consensus of those using something called AML-PRED. And when we looked at the consensus sequences that were aggregation prone and had the high propensity to aggregate if solvent exposed, they're shown in red here on the structure, and they're all buried. But the solvent accessibility of one of those in particular increased quite a lot as we went from pH 7 to 3.5. So we think that may be what's driving the increased aggregation kinetics at this pH, that region becomes more accessible, and therefore they can interact with each other and start to form aggregates. So what can we do about it is the next sort of question in terms of protein engineering. And we've, so Cheng Zhang has looked at a whole series of mutations, of which these are some shown, and really to try and modulate the dynamics and unfolding of these regions. And what he found was, particularly in the hinge region down in CL and CH regions, that, so it is particularly the CH in this case, that this region was very sensitive to mutation in terms of certain mutations were dampening down aggregation kinetics. And sort of coming back to the earlier message of does TM correlate with aggregation kinetics, well, what we found here was that the TM wasn't changing, even though the aggregation kinetics was slowing. And it turned out it was the entropy of unfolding that was important, which relates to how dynamic the initial structure is. If it starts off in lots of different conformations, got lots of conformational freedom, then you'll get a low delta S. And as you lock those down, you get a bigger delta S, which is the difference in entropy between the static native state and the much more extended unfolded structure. So it was about the dynamics of the protein that seemed to be more important rather than the overall unfolding propensity. We've also looked at binding excipients. So one way you can stabilize your protein is to add in sugars or amino acids. And this is work that's soon to be published. We're right on the edge of it, working with Paul Matishek at NIBS, and also Kate Groves and Milena Quaglia at LGC, using HDX mass spectrometry, where you can map regions of the protein structure at a peptide level that are interacting with the additives that you put in. And we could see the bits shown sort of the darker colors for each excipient, those regions that are responsive in terms of becoming more protected from the solvent. And that means the excipient is likely binding to, or is destabilizing these regions of the protein. And you can see they're in hotspots in particular regions, sometimes 3 hotspots, sometimes 2, depending on the amino acid. And that implicates them binding into those regions. So that's the beginnings also of some computational docking work that we've done on this, but also we do molecular dynamics simulations where we look at how the interaction of excipients and proteins evolves through a dynamic trajectory. And we've done this at a series of concentrations of glycine in this particular example. What we know about glycine is it stabilizes the protein. We get an increase in the melting temperature, so it conformationally stabilizes it, but it saturates, it stops improving it after a while. And what we found was actually glycine initially makes the protein more dynamic, which is sort of a bit counterintuitive to its stabilization. And we're just getting to the bottom of why that is. But then it becomes more rigid again. And then later on starts to become more dynamic again, because it's extremely complex. What's going on in the solution is very complex as well. So we're trying to piece together what this is telling us in terms of how it relates to the melting temperature. But it's certainly not a simple one thing causes another. There's complex interactions of things going on, which more will be revealed next time I give a presentation. So then up in Manchester on their side, they've been, so Robin Curtis has been looking a lot in terms of novel excipients, and he started off looking at dipeptides. So we know, in the example I've just shown you, that individual amino acids can be stabilizing. In particular, arginine is known to suppress aggregation, and glutamic acid is also known to stabilize proteins. So they looked at combinations, including phenylalanine, leucine, etc., and valine, as dipeptides to see if they could get any improvements through those. And so that fished out some examples, particularly arg-glu and arg-phe, which could modulate the aggregation kinetics of the protein. They've also been looking at something called sodium tripolyphosphate, which is a kind of, I guess, an analog of what you would get in ATP without the adenosine. It's known that ATP is stabilizing in cellular systems. So they looked at whether the tripolyphosphate element was responsible. And it's really interesting work. So they kind of looked at increasing the amount of STPP in solution with lysozyme. And they saw initially at a very small amount, you get protein precipitation, but as you then increase the amount into this, it overcharges the surface of the protein and allows it to go back into solution. So initially you form these kind of ionic salt bridges across proteins, so the STPP bridges the proteins and sticks them together, but eventually the net surface on the protein is so high that they colloidally repel each other. And that comes out in measurements of the zeta potential. And also he's looked at B22, which measures that interaction strength between proteins. And from this data as well, looking at the temperature at which it begins to increase the hydrodynamic radius, in other words, precipitate, he could see that as you added STPP in purple and also ATP, that suppressed the temperature to the point actually where you're not even getting large particles. So it was really suppressing the propensity of proteins to aggregate. Should just note that STPP is used in foods already, so it's potentially an excipient ready for use in pharmaceutical formulations. And then Jim Warrico, Manchester's looked at modelling this, and so he's modelled the STPP and its effect on the charge, net charge of the protein, and you can get a very good correlation between the zeta potential measured in terms of the charge on the protein, under different conditions versus what is experimentally modeled on the y-axis. So that's a really good correlation. It shows that it kind of confirms that it's a net charge of the protein surface, but also shows that we can model that. And then just looking at what Jim has done on top of this, one of the aims of this and related work with Jim Warwicker was to get a protein solubility server online, and I know he's done various bits of work on this from different sources like BioProNet. Really as a tool where you can put in your sequence and potentially structure of your protein and get a sense as to what its net charge is, what its solubility is. Are there any patches on there that you should worry about, and therefore you can go away and engineer them. And so this is just to point out that server is up and running. It's been up and running for a few years, I think. It has lots of functionality, of which some of them are kind of shown in these next slides. For example, this is calculating the polarity on the surface in terms of polar and non-polar residues, and this is the charge showing red in negative and blue positive charge on the surface. So you can see what kind of features on the surface might influence solubility, for example. Another thing he's been able to implement on this is to predict in terms of the dependence on pH and ionic strength. Compared to what we see experimentally, this is data for a fab, I think. So experimentally, we see that aggregation is a real problem at the low pH, higher salt. But there's a kind of a solubility point right down in this bottom left-hand corner. And that's related to the net charge on the protein. What he's got is the folded state stability predicted on the top here and the net charge on the bottom, and they together explain our experimental data that we typically see with fab molecules. So that's all a nice feature. He's also been using machine learning, taking an already published data set of 137 monoclonal antibodies where various measurements have been taken with them, and that's all in this publication here. So they've looked at the hydrophobicity as measured by hydrophobic interaction chromatography, and various other kind of responses in terms of solubility and what would be developability measures that you would use in industry. And there are 12 of those developability measures. I know there's now a 13th that's been added in a later paper. And what he's done is then use machine learning, which they didn't use, to predict where any given antibody would fit relative to this data set. And this is an introduced antibody, and it predicts its, in this case, the HIC hydrophobicity, or where I guess it's chromatography elution point versus the experimental value for that. And so you can get a good prediction with this method. He can also measure pKa's of surface residues. That's another tool on there. And so I've summarized all that together because there's lots of bits and pieces, I know, because we had a lot of aims here. But essentially, in terms of aim one, trying to understand factors affecting aggregation, we've gone a long way into understanding the role of local dynamics on unfolding, exposure of aggregation hotspots, APRs, and also that the net charge is a key feature that we need to, or you can use to stabilize proteins from aggregating through colloidal stability. So if you were aiming in formulation to improve the stability, you'd be looking at trying to increase the TM, definitely to a certain point, but also decrease the dynamics, so the flexibility of the molecule, and also increase its net charge. They're all tools you can potentially use to stabilize your protein. Can we predict better formulations? So it's all very well saying these are the things that need to change, but which are the most important for a given protein is very difficult to pick out. So we can predict dynamics and aggregation hotspots computationally. We can also look at where the excipients tend to bind through docking and also experimental methods. We can predict the net charge of the protein and the effect that excipients have on that. And as you can see, the server in Manchester is now designed to pull all that together and give you all that information together and potentially use machine learning then to understand it all collectively. What do we then engineer? Can we use that information to go on and engineer the protein or engineer the formulation? Well, we can certainly mutate the protein to suppress its dynamics, which we know has a benefit. Even if we're not increasing or needing to increase the melting temperature, it will decrease the aggregation kinetics. And also now we have more excipients, potentially based on these dipeptides and STPP, with which to stabilize our protein. And more excipients is definitely needed because it's quite a limited range that we can use in injectables. So with that, I shall just acknowledge all the various people. Certainly the key people are in the box in terms of who worked on each side of things at the different sites, both PDRAs, and we also aligned a number of PhD students into this. And there are lots of collaborators who fed in materials which are important, and obviously the funders. So there's mainly EPSRC funding here, but there's a bit from BBSRC on certain bits of the PhD project, for example. So with that, we'll be happy to take any questions if there is time.
Great, Paul. Thank you very much. You stuck to time nicely. We've certainly got time for one question, and I guess picking one out of the chat. The question was from Tim Ackerman, and it was, is it possible that rather than forming salt bridges between protein particles, the STTB is stabilizing an agglomeration of protein particles?
So this is Robin Curtis's area, and he's actually online. He might be able to answer for me. But so the STPP initially is precipitating or encouraging precipitation. I don't know if Robin wants to comment on it.
I mean, I think initially it follows the behavior of what's been observed in the literature with trivalent cations and proteins, and it definitely, there's some sort of attraction due to it binding, cross-linking proteins together, which causes them to precipitate, and then they resolubilize, and when they resolubilize, they're in monomeric form. So I'm pretty sure that it's not stabilizing an agglomeration of protein particles when it's in the solubilization regime, if that's what the question is.
I think Robin, that seemed to answer it. Thank you. Yeah, I mean, I think what Tim was suggesting was there's some stabilizing role, and it sounds like there isn't. It is the